Class 9, Semester 2. Chapter A, Food Biotechnology. Tempeh is a food ingredient made from fermented soybeans with the help of fungi and yeast. Tempeh is more easily digested by the body than soybeans and contains antioxidants that are able to ward off cancer-causing free radicals. Tempeh is one of the food products resulting from biotechnology because the manufacturing process involves microorganisms. Besides tempeh, what are some everyday foods that are products of biotechnology? Are the products of biotechnology only in the form of food? To find out the answer, let's study the following summary of the material. A. Definition of biotechnology. Biotechnology is the use of living things to help work or produce useful products for humans. Biotechnology began in 1857 after Louis Pasteur discovered the results of fermentation carried out by microorganisms. In the 1920s, fermentation involving microorganisms began to be used to make complex chemical solutions, such as the manufacture of alcohol. Microorganisms are living things that are very small and you have to use a microscope to see them. Utilization of biotechnology for the manufacture of food in everyday life. For example, in the manufacture of tempeh, tape, oncom, and so on. The development of food biotechnology then entered the era of modern biotechnology, which applies genetic, biochemical, and biomolecular principles. Modern biotechnology, such as the discovery of enzymes that help in the genetic engineering process, allows scientists to compile the genetic material of each organism to produce high-quality animals and plants, for example genetically engineered fish that are larger than normal fish. B. Conventional food biotechnology. Conventional food biotechnology is biotechnology that uses microorganisms as a tool to produce products and services. For example, fungi and bacteria produce certain enzymes for body metabolism so that the desired product is obtained. For example, tape is made by using microorganisms and yeast. Microorganisms convert organic substances into other organic substances, namely fermented cassava, into tape by using yeast, a type of fungus in yeast, namely Saccharomyces cerevisiae. During the process of making tape, the following is cassava and yeast tape. The picture is as follows: A. Cassava tape. B. Yeast Saccharomyces cerevisiae. There are three types of microorganisms in tape yeast, namely Aspergillus sp, Saccharomyces cerevisiae, and Acetobacter aceti. Microorganisms in tape work synergistically, meaning that microorganisms work alternately to convert raw materials from cassava or glutinous rice into tape. Following is the process of making tape. When making tape, starch is fermented into glucose assisted by Aspergillus sp, to convert glucose into alcohol assisted by Saccharomyces cerevisiae. To convert alcohol into vinegar is controlled by Acetobacter aceti. This mechanism of action is called synergism of glucose into alcohol. This process of change occurs because of the work of enzymes in yeast cells produced by microorganisms. Tape fermentation utilizes anaerobic respiration, breathing without oxygen, in microorganisms. Other examples of conventional food biotechnology are yogurt, cheese, tempeh, soy sauce, bread, and alcoholic beverages. Yogurt is the result of fermenting milk using Streptococcus thermophilus or Lactobacillus bulgaricus bacteria, which can convert lactose in milk into lactic acid. The breakdown of protein in milk causes milk to become thick and tasted. This decomposition process is called lactic acid fermentation, and the end result is yogurt. Cheese is a food ingredient that is produced by separating solids in milk through a thickening process, coagulation, carried out with the help of bacteria Lactobacillus bulgaricus and Streptococcus thermophilus, which can produce the enzyme renin, so that the protein in milk will coagulate and divide the milk into liquid and solid curd. Then the enzyme renin converts the lactose sugar in milk into acids and proteins in the curd. Curd is further processed through a ripening and packaging process to form a processed food called cheese. Tempe is a traditional Indonesian food that is fermented. Fermentation is done by growing the fungus Rhizopus oryzae and Rhizopus oligosporus on soybean seeds. In the process of growth, fungi produce threads called hyphae. Hyphae cause soybean seeds to bind together and form a compact structure. The following is an example of Tempe and Rhizopus bacteria. Tempe. B. The fungus Rhizopus oryzae. When the fungus grows, the fungus makes a protease enzyme that can break down the complex protein in soybeans into amino acids that are more easily digested by the human body. The following is the nutritional content of soybeans and tempeh. Soy sauce is a biotechnology product made from soybeans. Soybeans were fermented using the fungus Aspergillus wendtii. Then the fermented soybeans were dried and soaked in a salt solution. Making soy sauce is done through the process of soaking soybeans in a salt solution. So making soy sauce is called salt fermentation. The fungus Aspergillus wendtii breaks down protein into amino acids, components of taste, acid, and distinctive aroma. Bread making utilizes yeast-assisted fermentation, namely Saccharomyces cerevisiae. Yeast is a fungus that is added to flour dough and causes a fermentation process that produces CO2 gas and alcohol. CO2 plays a role in developing bread. Alcohol produces aroma and gives bread its taste. The dough will appear to expand when you put it in the oven, because the gas will expand at high temperatures. Alcoholic beverages. The manufacture of alcoholic beverages using fermentation with the help of the fungus Aspergillus oryzae. Aspergillus mushroom oryzae produce the enzyme amylase, which can break down starch into glucose or sugar. Furthermore, the sugar is further fermented into alcohol and carbon dioxide gas. Then produce alcoholic beverages with a certain taste according to the raw materials used. The longer the fermentation process, the higher the alcohol content. Examples of alcoholic beverages are wine or wine. Wine is made from grapes by utilizing Saccharomyces cerevisiae through a fermentation process, as well as fermentation in the manufacture of alcohol in general. However, consuming alcohol will damage health. The government limits the consumption of alcohol through the Food and Drug Administration (BPOM) by recommending a limit of alcohol on food and beverages as much as 5%, if more than 2%. BPOM declares the food and drink dangerous to health. Modern food biotechnology. Modern biotechnology in food production is carried out by applying genetic engineering techniques. Genetic engineering is the activity of manipulating genes to get new products by making new DNA by adding or removing certain genes. An example of a genetically modified product is making a transgenic organism. Through genetic engineering, biotechnologists can arrange gene patterns so as to produce organisms whose characteristics are as needed. This technique is called recombinant DNA, which is the process of combining DNA from one organism to another. Genetic patterning involves the use of another organism's genes that are inserted into a particular organism's DNA band. Organisms that use parts of the genes of other organisms in their bodies are called transgenic organisms. Transgenic plants, animals and bacteria are not only used for research purposes but also to meet needs in the medical and agricultural fields. Transgenic plants are plants that have changed the composition of their genetic material. Transgenic plants are an alternative so that plants are resistant to pests so that yields are abundant. Plants can be engineered to be able to kill pests that attack plants. In 2003, about 67.7 million hectares planted by 7 million farmers in 18 countries began to plant transgenic crops, namely soybeans and kapok plants that have herbicide and insecticide resistance. The following is a picture of a plant genetic engineering technique with the help of Agrobacterium tumefaciens. Genetic engineering techniques are carried out through 4 stages, namely, 1. Prepare DNA fragments that are inserted into the DNA of certain plants. 2. Prepare vectors intermediaries using plasmids or viruses. 3. The pieces of DNA to be inserted or recombined with vectors. 4. Recombinant DNA is inserted into plant cells. 5. The plant will grow into a plant with new properties according to the inserted DNA. Through GMOs, peanuts and soybeans are also developed without causing allergies to those who consume them. The production of transgenic plants that pass the field test can be marketed freely. Another GMO crop is virus-resistant sweet potato. Rice containing high iron and vitamin A is called golden rice. Here's an example of golden rice. Researchers carried out genetic modifications to produce and accumulate provitamin A, beta-carotene. Provitamin A was converted into vitamin A when a plant is injured. A collection of cells called callus grows faster at the injured site. Callus has the ability to differentiate into stems, shoots, roots and all organs of flowering plants. The natural potential for reprogramming makes callus expandable through genetic engineering. Callus is also covered by thick cellulose on its cell walls so that it can become a barrier or inhibit new DNA that will enter. The cell wall can be broken down with the help of the cellulase enzyme, resulting in a cell without a wall called a protoplast. Protoplasts can be fused combined with protoplasts from other plants, resulting in cells that can grow into a hybrid plant. The method is called protoplast fusion. This method has been used on broccoli, which is the result of the fusion of broccoli plants with cauliflower plants. Here is a broccoli plant. d.Animal biotechnology. Transgenic animals were originally the subject of research by scientists to find diseases that attack certain animals and how to overcome them. Furthermore, animal genetic engineering aims to produce farm animals that produce quality milk and meat, fish that grow quickly and contain certain vitamins. The dairy industry is a target for the increase in GMOs. Researchers are using GMOs to increase milk production by making milk rich in protein, low in fat, and better for human consumption. Researchers carry out genetic engineering on farm animals, so as to be able to develop livestock that are resistant to disease. In addition, transgenic organisms are currently being developed that can be used as a source for making organs that can be used as organs in the transplant process. Biotechnology is also used for reproductive engineering in animals such as the cloning process. Cloning is copying one individual into another physically and genetically identical individual. Cloning is asexual reproduction that aims to produce a new, uniform individual. Cloning is also used to obtain superior animals and plants. The following illustrates the cloning process in sheep. How to clone sheep A. That is, first take some body cells from sheep A, then take egg cells from sheep B. Cells from sheep A's body are taken only the cell nucleus, and egg cells from sheep B are taken the cell nucleus so that the egg cell body remains. The nucleus of the sheep A cell is injected into the egg cell of the B sheep. Furthermore, these cells will develop into an embryo and be implanted in the womb of another sheep, sheep C. After that, sheep that are similar and identical to sheep A will be born. The lambs that are born have characteristics that are identical to the cell nucleus donor sheep. In this illustration, the A sheep cell nucleus donor. Every GMO product on the market is given a special label that indicates GMO food ingredients. GMO products are also widely used in the medical field, for example, the manufacture of the hormone insulin through bacteria. Insulin is a hormone produced by the pancreas, which plays an important role in the digestion of carbohydrates. Insulin is used to treat patients with diabetes mellitus. e.Impact, a biotechnology. Scientists continue to conduct research in the field of biotechnology that can produce new products that can improve human welfare. However, biotechnology also causes losses in three fields, namely the environmental, health, social and economic fields. Environmental sector, transgenic plants or animals have a modified gene arrangement, namely the addition or subtraction of the organism's genes. These transgenic organisms, if not managed properly, can contaminate the diversity of genes in the natural environment or damage the germplasm, which is called gene pollution. For example, herbicide-resistant maize plants. When planted in natural land, pollen can carry the genetically modified maize gene and pollinate natural maize. This makes the genes in natural corn contaminated with genes from transgenic corn. GMO crops are usually superior crops. This makes farmers tend to plant GMO crops monoculture and no longer grow local crops. As a result, local plants become scarce and the amount of germplasm decreases. In addition, it creates new pests that are stronger than the previous pests that can disrupt the ecosystem. Health sector. In transgenic organisms there are foreign genes that should not exist even for human consumption. This gene is feared to trigger the emergence of new diseases or even cancer. Based on the results of research, transgenic soybean plants containing Brazil nut genes can trigger allergies in people who are sensitive to Brazil nuts. It is feared that foreign genes can trigger resistant bacteria so that more virulent bacteria appear. Examples of biotechnology products such as alcohol can be misused to make alcoholic beverages which, if consumed continuously, can have a negative impact on health. social and economic sector, biotechnology products also affect the social and economic fields. People who have the capital can develop GMO agriculture that can increase yields to become abundant with very good quality. This makes traditional farmers unable to compete in marketing to the detriment of traditional farmers. If it continues, it will lead to a bigger economic gap. Likewise, developed countries have developed transgenic organisms that market their products in international trade, making developing country products will lose so that the country's income will decrease. This makes developing countries become dependent on the products of developed countries.